Okay, let's unpack this. Have you ever considered, really thought about, what gives your tissues that remarkable resilience?
Mm.
Or, that amazing elasticity in your skin, or even just the slippery glide in your joints?
Yeah, things we take for granted.
Exactly. And what allows cells to recognize their neighbors or even fend off invaders, there's this incredible unseen complexity within our bodies.
Right.
It's like a foundational scaffolding known as the extracellular matrix, and it underpins so much of our physical being.
It really does. It's everywhere.
So today, we're embarking on a real deep dive into three crucial families of biomolecules. They're the true architects of this matrix, shaping our bodies, keeping them running. We're talking glycosaminoglycans, or GAGs, proteoglycans, and glycoproteins.
Big names, but fascinating stuff.
Definitely. And this isn't just about learning some, intimidating scientific words. It's about peeling back the layers. We want to understand the fundamental architectural wonders that literally allow you listening to move, think, and interact with the world.
Absolutely.
We've brought together a really comprehensive stack of source material for this. It's a truly expert look at these biomolecules, designed to give you the deep knowledge needed to truly master this field. We're going beyond the basics here.
Yeah, and what's truly fascinating, and what we're going to really explore in depth today, is how these seemingly abstract molecular structures, how they translate directly into the actual physical properties we experience every single day.
Right, the tangible stuff.
Exactly. From the clarity of your vision, the flexibility of your joints, even how your immune system functions, we're going to connect these molecular dots.
Oh,
How they're built and how they're broken down. We'll link that to the bigger picture of human health and disease.
And crucially.
And crucially, yes, we'll examine what happens when these incredibly intricate processes go wrong. The goal here is a really comprehensive understanding, something that genuinely approaches mastery of the subject.
Excellent. Let's get into it then. So let's start our journey with glycosaminoglycans, or GAGs. The name itself might sound like a bit of a mouthful.
It does, yeah.
But their design and function are, incredibly elegant,
Well,
really. You can think of them as these unsung heroes providing this hydrated scaffolding within our bodies. But what exactly are they at their core?
Okay, so at their most fundamental level, GAGs are large, complex molecules. They're made of what we call negatively charged heteropolysaccharide chains.
Heteropolysaccharide. Okay, break that down a bit.
Right, so hetero meaning different, and polysaccharide meaning many sugars. So these aren't just simple chains of one repeating sugar unit.
Oh,
Instead, they're chains built from two different kinds of sugars that repeat over and over, which gives them this unique complexity. And here's where their power really starts. They are overwhelmingly negatively charged.
that negative charge sounds important.
It's absolutely critical. Generally, these GAG chains are found associated with a small amount of protein. We call that the core protein. And when these GAGs link up with that core protein, they form molecules called proteoglycans. We'll definitely dig into those soon.
Right.
What's amazing about these proteoglycans is just how carbohydrate-rich they are, sometimes up to 95% carbohydrate by weight.
Wow, that's mostly sugar then.
Pretty much. But the absolute most critical property of GAGs, the thing that really unlocks their functional power, is their extraordinary ability to bind really, really large amounts of water.
Like a molecular sponge you mentioned earlier.
Exactly. Think of a molecular sponge. This impressive water binding capacity is precisely what allows them to form that resilient, gel-like matrix. That's what makes up the ground substance of our body.
Ground substance. So that's like the filler between cells.
Kind of, yeah.
Yeah.
You can picture this ground substance as the vital filler material surrounding cells and tissues. It provides support, but also a medium for transport. And this ground substance, along with key fibrous structural proteins, things like collagen for tensile strength.
Right, collagen's a big one.
Elastin, which gives elasticity, and fibrillin-1, providing structure for elastic fibers, plus adhesive proteins like fibronectin. All of that together forms the extracellular matrix, the ECM.
Okay, so GAGs are a key part of this ECM structure.
Absolutely fundamental. Within this ECM, the hydrated GAGs play multiple vital roles. First, flexible support. Think shock absorbers, giving the ECM resilience, cushioning. They're constantly interacting with those other proteins.
Got it.
Second, they act as a kind of molecular sieve.
A sieve? How so?
Well, they meticulously influence how materials like nutrients or waste products move through the ECM. They essentially act as a selective filter for molecular traffic.
Interesting. Okay.
And finally, maybe one of their most recognizable roles, though often unseen, is contributing to the viscous and lubricating properties of things like mucus secretions.
Like in our airways or gut.
Exactly. That's why, historically, they were actually called mucopolysaccharides.
Makes sense.
It's incredible how these seemingly simple properties, just negative charge and water binding, lead to such diverse and critical functions throughout the body.
It truly is. It's easy to just dismiss them as filler, but realizing they're active filters and shock absorbers, that really changes the perspective. So how did they pull this off structurally? What is it about their atomic architecture that makes them so suited for these roles, especially that incredible water binding?
That's where we get to the heart of their elegance, really. GAGs are characterized by these long, unbranched heteropolysaccharide chains.
Unbranched, okay, so just straight lines.
Pretty much, yes. This linear, unbranched structure is crucial for their function. It lets them extend out and occupy space effectively. They are built from repeating disaccharide units.
Disaccharide, meaning two sugars.
Exactly. A two-sugar unit that repeats hundreds, maybe thousands of times over, like beads on a very long, very specific necklace. And each of these repeating units always consists of two main components, an acidic sugar and an amino sugar.
Okay, acidic and amino. Let's break those down.
Right. So for the amino sugars, you're typically looking at either D-glucosamine or D-galactosamine. Now, here's a key detail for mastery. The amino group on these sugars is usually acetylated.
Acetylated, why is that important?
Well, this acetylation neutralizes what would otherwise be a positive charge. It helps the molecule maintain its overall negative character.
Ah, I see. Keeps it negative.
Precisely. These amino sugars can also be further modified by being sulfated. That means a sulfate group gets attached, maybe on carbon 4 or 6, or sometimes even on a non-acetylated nitrogen. And those sulfate groups, of course, carry a strong negative charge.
More negative charges.
And then you have the acidic sugar component. This is usually D-glucuronic acid, or it's C5 epimer L-iduronic acid. These uronic sugars are special because they contain carboxyl groups.
Carboxyl groups, like in acids.
Exactly. And at physiological pH, the normal pH of our bodies, these carboxyl groups are negatively charged. So you combine the negative charges from these carboxyl groups with the negative charges from the sulfate groups.
And you get something incredibly negative.
You got it. GAGs are overwhelmingly negatively charged. This strong negative nature is the secret to their function. It's also worth noting one unique exception: keraton sulfate.
Inception.
Yeah. Unlike the others, keraton sulfate contains galactose instead of an acidic sugar in its repeating unit. But for the vast majority of GAGs, that strong negative charge from carboxyl and sulfate groups is the defining characteristic. Really nail that down for understanding them.
Okay, this is where it gets really interesting for me, and I think this is a true aha moment for understanding how our bodies work mechanically. How do these negative charges, this seemingly simple molecular property, lead to such vital properties like slipperiness and resilience? It's not immediately obvious how, molecules pushing each other away translates into cushioning your joints or giving your eye its shape. Help me wrap my head around that connection.
That's the absolute core principle of GAG function, and it's just beautifully elegant in its simplicity.
Yeah.
Because these heteropolysaccharide chains are so heavily negatively charged, they naturally repel each other.
Like magnets pushing away.
Exactly like trying to push together two magnets with the same polarity they push away, right? GAG chains do the same thing in solution. This causes them to extend outwards to occupy a large volume, but it's not just the repulsion between the chains themselves.
Okay.
Each GAG chain is also surrounded by a shell of water molecules. Water is highly attracted to those negative charges, forming a tightly bound hydration layer around the GAGs. This water layer is absolutely crucial.
The water binding again?
Yes. Now let's think about slipperiness. When GAGs, like the ones in mucus secretions or the synovial fluid lubricating your joints, are brought together, their strong negative charges, combined with those surrounding water shells, allow them to just slide past each other with minimal friction.
Wow.
This is exactly what gives substances like mucus and synovial fluid their incredibly slippery, lubricating consistency. It's like molecular ball bearings cushioned by water.
That's a great analogy.
Okay.
What about resilience then?
Right, resilience. Imagine a tissue or fluid containing GAGs, say, the vitreous humor filling your eye, giving it its shape, or the fluid in your joints.
Okay.
When the solution gets compressed, maybe you put pressure on your joint by standing up or moving the water, gets temporarily squeezed out from around the GAGs. This forces them to occupy a smaller volume for a moment.
Right, they squish down.
But here's the magic. The gag chains themselves, with their constant negative charges, still strongly repel each other. So as soon as that compression is released, boing,
They spring back.
they spring back. Those gags want to return to their original extended hydrated volume because of that powerful repulsion between their negative charges. This spring back property is fundamental to the resilience, the shock absorbing capabilities, the shape maintaining functions of tissues and fluids all over your body.
So cushioning joints, keeping the eye shape, skin integrity.
Exactly. It's an elegant self-recovering system, all driven by charge and water.
So we have this elegant, negatively charged, water-binding structure providing incredible resilience and lubrication. Given how many different roles they play, are all GAGs the same? Or do they come in different flavors for different jobs, maybe subtle variations in structure?
Oh, they absolutely come in different flavors. Each one is exquisitely tailored for specialized roles and specific locations in the body. There are 6 major types of GAGs, generally speaking.
6 types, okay.
And they're classified based on a few distinct characteristics. Things like their monomeric composition, which specific sugars make up their repeating units, the type of glycosidic linkages connecting those sugars, and also the degree and precise location of their sulfate units. Even minor differences here can lead to significant changes in function.
Details matter.
Immensely. And here's a really important distinction to remember, especially if you're aiming for mastery. All GAGs, with one single crucial exception, are sulfated.
Okay, one exception. Which one?
That exception is hyaluronic acid.
Hyaluronic acid, right. I've heard of that.
Very common, yes. And critically, most GAGs are also found covalently attached to a protein, forming those proteoglycan monomers we mentioned briefly. We'll definitely dive into those next.
Oh, yeah.
The key examples of these major GAG types, each with its own unique distribution and functions, include chondroitin 4 sulfate and chondroitin 6 sulfate, vital for cartilage.
Right.
Keratin sulfate, also in cartilage in the cornea. Dermatin sulfate, found in skin, blood vessels, heart valves. Heparin, a powerful anticoagulant.
Heparin, yeah.
Heparin sulfate, found on cell surfaces and in the ECM, involved in cell signaling. And, as we noted, hyaluronic acid, unique because it's unsulfated and not typically covalently linked to protein, plays a huge role in joint lubrication and wound healing. Each one fulfills a vital, specialized role. It just showcases the incredible precision of molecular design.
It's clear GAGs are amazing on their own, giving tissues that resilience and slipperiness. But you mentioned they often link up with proteins to form proteoglycans, and I love that bottle brush analogy you used. How does adding a protein to that mix change their function or, allow them to do even more complex jobs in our bodies?
It's a perfect progression, really, because the proteoglycan structure takes the basic power of GAGs and just amplifies it. It adds another layer of intricate organization, and that bottle brush analogy is excellent, yeah.
Yeah, it really helps visualize it.
It visually captures both their structure and how they operate. Proteoglycans are primarily found in the extracellular matrix, that elaborate network surrounding our cells.
Right, the ECM again?
Exactly. Where they provide structural support, signaling capabilities. They do a lot. They're also found on the outer surface of cells, playing vital roles in cell-to-cell communication and recognition.
So structure and communication.
Precisely. Let's dissect their monomer structure. A typical proteoglycan monomer, and these are found in incredibly high abundance, especially in cartilage, consists of a central core protein. To this core protein, anywhere from a few up to 100 linear chains of GAGs are covalently attached.
Uncovalently, so a strong bond.
Very strong, stable chemical bond. Each of these individual JAG chains can be incredibly long, sometimes up to 200 disaccharide units.
Wow.
And here's why the bottle brush analogy holds. Because of their strong negative charges, these GAG chains extend outwards radially from that central core protein. They naturally repel each other, maintaining separation.
Ah, like the bristles on a brush.
Exactly. This radial arrangement of the bristly JAG chains around the central protein is precisely what gives them that characteristic bottle brush look. In cartilage, proteoglycans specifically, the main GAGs attached are typically chondroitin sulfate and keratin sulfate.
Right, the ones you mentioned are key for cartilage.
Yes, and they both contribute significantly to cartilage's unique load-bearing and lubricating properties. It's also worth noting, for a deeper understanding, that proteoglycans are organized into gene families. These families encode core proteins with common structural features.
Like the aggregan family.
Exactly. The aggregan family includes aggrecan itself, which is super abundant in cartilage, plus versican, neurican, brevican, each playing specialized roles in different tissues.
Okay. So how does the JAG actually link to the protein? Is it just stuck on anywhere?
Oh no, it's much more specific than that. It's a fascinating piece of molecular engineering. It's not just a simple attachment, it's a very specific chemical bond. Most commonly, this covalent linkage happens through a special trihexoside linker.
Trihexoside, 3 sugars.
Yes, a 3-sugar unit made of galactose-xylose. This linker is then attached to a serine residue on the core protein. Specifically, an O-glycosidic bond forms between the xylose sugar of the linker and the hydroxyl group of the serine amino acid.
Glycoprotein. Okay.
It firmly anchors the JAG chain to its protein backbone. This stable linkage ensures the GAGs stay precisely where they're needed.
Okay, so we have these bottle brushes. Is that the whole story?
Not quite. The complexity and, frankly, the impressiveness continues with aggregate formation. Many of these individual proteoglycan monomers, these bottle brushes, don't just float around alone.
No.
Especially in tissues like cartilage, they can non-covalently associate with one single very large molecule of hyaluronic acid.
Wait, they link up with hyaluronic acid too, the unsulfated one.
The very same.
Ooh.
This creates truly massive structures, gigantic complexes. This association isn't a strong chemical bond like the JAG protein link, but rather occurs primarily through crucial ionic interaction.
Ionic. So charge attraction.
Yes, between the core protein of the proteoglycan monomer and the long hyaluronic acid backbone. And to further stabilize this enormous aggregate, additional smaller proteins called link proteins get involved.
Link proteins, okay, like molecular glue.
Kind of like molecular clamps, yeah. They hold the entire assembly together. This creates an incredibly vast, intricate, and superhydrated network. And this network is absolutely fundamental to the structural integrity and the remarkable shock-absorbing capacity of tissues like cartilage. It allows them to withstand significant mechanical stress.
That is truly an intricate level of organization. From individual sugars linking to a core protein to these massive aggregates held together by ionic forces and helper proteins. It sounds like a masterpiece of biological engineering.
It really is.
Okay, now that we know what they are, how they're structured, and these incredible properties they have, the biggest puzzle for me is how a cell, which is basically a tiny chemical factory, manages to build these complex multi-sugar chains with such precision, one sugar at a time. How does that monumental task even begin?
That's exactly where the real marvel of cellular engineering comes into play. Imagine an incredibly precise assembly line where every single step is meticulously controlled. When we talk about the general principles of JAG and proteoglycan synthesis, the basic idea is that these heteropolysaccharide chains get elongated by adding alternating acidic and amino sugars one after another.
Sequentially.
Sequentially, yes. The crucial donors for these sugars are primarily their activated forms. Often these are their uridine diphosphate or UDP derivatives.
UDP, like an energy tag.
Sort of, yeah. Think of them as sugar molecules with the special energetic tag that makes them ready to be linked onto the growing chain. These reactions are meticulously catalyzed by a family of highly specific enzymes called glycosyltransferases.
Right, enzymes doing the work.
Each one is designed to add a particular sugar at the correct position and using the correct type of linkage. And because GAGs and proteoglycans are ultimately destined for export out of the cell to become part of the ECM or function on the cell surface, their synthesis happens primarily within the Golgi apparatus.
The Golgi, the cell's post office, basically.
Pretty much. It's the cell's sophisticated processing, sorting, and packaging center for proteins and lipids that are heading out.
Okay, so let's start with the building blocks. Those amino sugars you mentioned earlier, how are they made?
Right. Let's delve into amino sugar synthesis first. They are truly essential components of many different glycoconjugates, not just GAGs, but also glycoproteins and glycolipids, as we'll see later.
So widely used.
Incredibly widely used. The synthetic pathway for these amino sugars, also called hexosamines, is super active, particularly in connective tissues. In fact, a significant chunk, maybe up to 20% of the glucose flowing through these tissues, actually gets shunted into this pathway.
Wow, 20%. That highlights its importance.
It really does. So how does the cell build these specific amino sugars like N-acetylglucosamine, GLC-NAC, and N-acetylgalactosamine, GalNAc? It starts with a common sugar, fructose 6-phosphate.
Okay, familiar starting point.
Yeah. Through a series of incredibly precise enzymatic reactions, think molecular assembly line, again, the cell strategically adds, removes, reconfigures atoms. A key initial step involves swapping a hydroxyl group on fructose 6-phosphate for the amide nitrogen from a glutamine molecule.
Okay.
The resulting glucosamine 6-phosphate product then undergoes more modifications. It gets acetylated, isomerized, and finally activated. This whole process results in the nucleotide sugar UDP-GLCNAC,
The activated form.
the ready-to-use activated building block. And then UDP-GalNAc, the other critical amino sugar, is generated by an epitrization reaction directly from UDP-GLCNAC, basically a rearrangement of its chemical structure. It's these activated forms, UDP-GLCNAC and UDP-GalNAc, that are directly used to elongate the growing carbohydrate chains.
Got it. What about that other one you mentioned, NANA, N-acetylneurominic acid?
Ah, yes, NANA. That's a fascinating molecule. It's a 9-carbon acidic monosaccharide and a prominent member of the sialic acid family.
Sialic acids, okay.
These compounds are typically found right at the very ends, the terminal residues of oligosaccharides side chains on glycoproteins and glycolipids. Less frequently on GAGs. They often act as important recognition markers on the cell surface.
Like little flags.
Kinda, yeah. The immediate precursors for nanosynthesis are N-acetylmonosamine 6-phosphate, which comes from fructose 6-phosphate, and phosphenol pyruvate, an important intermediate from glycolysis.
Okay.
Now, before NANA can be incorporated into a growing chain, it also has to be activated. This activation happens when it reacts with cytidine triphosphate, CTP, to form cytidine monophosphate NANA, or CMP NANA. This is catalyzed by the enzyme CMP NANA synthetase.
CMP NANA. Wait, what? CMP, not CDP or UDP.
Exactly. And here's a truly fascinating detail, one that's a huge clue for researchers and really highlights a unique regulatory mechanism, something key for mastery here. CMP NANA is the only nucleotide sugar in human metabolism where the carrier nucleotide is a monophosphate rather than a diphosphate.
Why? Why break the pattern?
That's the million-dollar question. It's a significant departure from the usual biochemical pattern. It suggests a unique regulatory need, maybe making NANA's pathway distinct, perhaps a powerful control point. It's not just a quirk. It's telling us something important about how NANA is handled by the cell.
Wow. So the cell not only builds these incredibly complex chains, but uses a special rule-breaking carrier for NANA, signaling its unique importance. That's a profound insight. What about the acidic sugars, like glucuronic acid? How are they synthesized and prepped for GAGs, and do they have any other surprising roles?
Excellent question, as they are equally crucial and definitely have broader implications. D-glucuronic acid and its C5 epimer, L-iduronic acid, are essential components of GAGs, as we said.
Right.
But glucuronic acid also plays a vital role beyond GAG synthesis. It's required for detoxification.
Detox. How?
Well, it helps get rid of various lipophilic or fat-soluble compounds in the body. Things like bilirubin, that breakdown product of heme, various steroids, and many common drugs, including statins. These are made more water-soluble by a process called glucuronidation.
Glucuronidation.
Yes, it involves conjugating or attaching them with glucuronate. This increases their solubility, making it much easier for the body to excrete them, preventing toxic buildup.
Super important function then.
Absolutely vital. While small amounts of glucuronic acid can come from our diet or from recycling existing GAGs through lysosomal degradation,
Right.
the major synthesis rite is actually called the uronic acid pathway. This pathway starts when glucose 1-phosphate reacts with UTP to form UDP glucose. This UDP glucose is then oxidized, producing UDP glucuronic acid.
The activated form again.
Exactly. That's the specific activated form used for both GAG synthesis and for those vital glucuronidation detox reactions. It's also quite interesting, though, maybe a bit of trivia.
Go on.
In plants and most mammals, notable exceptions being guinea pigs, primates, and of humans, glucuronic acid is actually a precursor for vitamin C, ascorbic acid.
Huh. So they make their own vitamin C that way.
Many organisms do, yeah, we can't. Furthermore, this uronic acid pathway provides a clever way for dietary D-xylulose, a less common sugar, to enter our central metabolic pathways, ultimately feeding into glycolysis.
Okay. What about the other acidic one, L-iduronic acid?
Right, L-iduronic acid. Its synthesis follows a different and somewhat counterintuitive timeline. It doesn't get synthesized as a free nucleotide sugar before it's put into the chain like most other sugars do.
No. How them?
Instead, the apomerization, that specific rearrangement flipping D-glucuronic acid to L-iduronic acid, happens after the D-glucuronic acid has already been incorporated into the growing carbohydrate chain.
While it's already attached.
Exactly. This transformation is catalyzed by an enzyme called uranosyl 5 epimerase. It flips the configuration from D to L sugar right there on the chain. This is a critical modification that dictates the final properties of certain GAGs like dermatan sulfate and heparin sulfate.
So we have all these activated sugar components, painstakingly prepared. But what about the protein part of the proteoglycan, the core protein? Where does that come into the picture, and how do these distinct sugar and protein bits get assembled into that complex bottle brush structure? Sounds like a master coordination effort.
It absolutely is a masterclass in cellular coordination. The core protein for proteoglycans, just like other proteins destined for secretion or membranes, is initially made by ribosomes attached to the rough endoplasmic reticulum, the RER.
Okay, made on the RER.
Right. Once synthesized, this protein enters the RER lumen, the internal space where it starts to fold correctly. From there, it moves on to the Golgi apparatus.
To the post office.
Exactly. And it's within the Golgi that this core protein truly begins its glycosylation journey, getting its sugar chains added by specialized membrane-bound glycosyltransferases.
Okay, so the sugars get added in the Golgi. How does it start?
The carbohydrate chain synthesis doesn't just begin anywhere on the core protein. It's very specific. It starts with the synthesis of a short linker region right on the core protein. This serves as the stable foundation for the rest of the long JAG chain.
A specific starting point.
A very specific starting point. The most common linker is that trihexoside we mentioned, the 3-sugar unit. This is formed when a xylose molecule, donated from UDP xylose, is transferred to the hydroxyl group of a specific serine or sometimes threonine residue on the protein. This initial critical reaction is catalyzed by xylosyltransferase.
Okay, xylose first.
Xylose first. Then, 2 galactose molecules are added to complete this trihexoside linker. Only after this specific linker is fully established does the sequential addition of the alternating acidic and amino sugars begin, building out that remarkably long JAG chain.
Got it.
And as we mentioned earlier with L-iduronic acid, it's during this elongation phase that the epimerization of some D-glucuronal residues to L-iduronal residues also takes place, fine-tuning the JAG's final structure and function right there on the assembly line.
Amazing precision. What about the sulfate groups? When did they get added?
The sulphation. That's a crucial final step. The sulphation of a JAG is not an early event. Instead, it occurs after the monosaccharide that's going to be sulfated has already been incorporated into the growing carbohydrate chain.
Added later then. Where does the sulfate come from?
The source of the sulfate for these reactions is a high-energy molecule called 3-minute phosphodinosyl 5-phosphosulfate, or PAPS.
PPS. Okay.
Think of PAPS as an adenosine monophosphate molecule with a sulfate group conveniently attached to its 5-phosphate. This makes the sulfate group highly reactive, ready for transfer.
Got it. And enzymes do the transfer.
Exactly. The actual sulfation reaction transferring the sulfate group to the JAG is catalyzed by a family of enzymes known as sulfotransferases. Each one is specific for a particular sugar residue and a particular position on that sugar.
So specific sulfotransferases for specific spots.
Precisely. For example, in making chondroitin sulfate, this final sulfation step is absolutely critical for its function in cartilage. And here's a vital clinical connection, really key for mastery. A defect in the sulfation of these growing GAG chains can lead to a group of devastating autosomal recessive disorders called chondrodystrophies.
Chondrodystrophies, affecting cartilage and bone.
Profoundly affecting skeletal development and maintenance, yes, leading to severe bone and cartilage abnormalities. It truly shows just how critical each and every step in this incredibly intricate synthesis pathway is for our health. One mistake can have huge consequences.
That's an astonishing amount of precision just to build these molecules. But as you said, it's a constant cycle in our bodies, creation and degradation. So building these complex molecules is one massive challenge, but breaking them down efficiently, that's just as vital, isn't it?
Absolutely crucial.
And when that degradation process goes wrong, we see some really profound health issues. Where does this breakdown happen, and how does the cell manage such a complex disassembly? Seems just as hard as building them.
You've hit on a critical point. The degradation of GAGs is a tightly regulated, highly specific process, and it occurs almost exclusively within the lysosomes.
Lysosomes.
Yeah.
The cell's recycling centers.
Exactly. Think of them as specialized recycling centers or digestive organelles. They contain a specific set of hydrolytic enzymes that use water to break down molecules. These enzymes are most active at an acidic pH, typically around pH 5. That's why they're collectively called acid hydrolases.
Acidic environment? Is that important?
Very. This low pH optimum inside the lysosome is actually a brilliant protective mechanism. It ensures that if these potent enzymes accidentally leak out into the cytosol where the pH is neutral,
They will work.
exactly, They won't function effectively, so they won't just start destroying the cell. It's a built-in safety switch.
Clever. How long do GAGs usually last?
Well, their half-lives, how long they typically last before breakdown, vary considerably. Depends on the specific JAG type, its location. Could be minutes for some signaling molecules or months for structural components.
Okay, so how do GAGs, which are mostly outside the cell, get into the lysosome for breakdown?
Great question. Since GAGs are primarily extracellular or on the cell surface, they can't be degraded right where they are. The very first step involves them being engulfed by the cell.
Engulfed?
Yes, through a process called phagocytosis, or sometimes endocytosis. The cell membrane invaginates, folds inward, forming a vesicle that encloses the GAGs.
Like Pac-Man eating them.
Sort of like that, yeah. This newly formed vesicle containing the GAG then fuses with the lysosome. This creates a single, larger digestive vesicle where the GAGs can be efficiently broken down in that acidic environment.
Okay, so they get brought inside and delivered to the lysosome. What happens then?
The complete lysosomal degradation of GAGs requires a large number of these specific acid hydrolases. Each one has its own precise job. It's not a random demolition. It's a highly ordered disassembly.
Ordered. How so?
First, the long polysaccharide chains get chopped up into smaller fragments by enzymes called endoglycosidases. These cut within the chain.
Okay, initial big cuts.
Right, producing smaller oligosaccharides. Following that, the further degradation of these oligosaccharides happens sequentially, starting from the non-reducing end of each chain.
The non-reducing end, like
Yeah.
dismantling a necklace one bead at a time from one end.
Exactly like that. And there's a general rule here, often called the last on, first off rule.
Last.
On, first off. Yeah. The last group, whether it's a sulfate or a specific sugar that was added during the GAGs synthesis, is typically the very first group that gets removed during degradation.
Wow, so it's undone in reverse order?
Precisely. This removal is carried out by highly specific enzymes. Sulfatases remove sulfate groups, and exoglycosidases remove individual sugar residues from the end of the chain. It's also worth noting these endo and exoglycosidases aren't exclusive to GAGs. They're also involved in breaking down glycoproteins and glycolipids. The critical and often devastating consequence of deficiencies in any one of these specific degradative enzymes is the pathological accumulation of partially degraded carbohydrates within the lysosomes. This buildup leads to significant tissue damage, cellular dysfunction, and a whole array of severe clinical symptoms.
That last on, first off rule is fascinating. Such precise enzymatic control, like a perfectly choreographed demolition. But when that choreography breaks down, especially in GAG degradation, it leads to a very specific and often devastating group of inherited diseases. Can you tell us about the mucopolysaccharidoses, or MPS?
Yeah.
What have we learned about them through this deep dive?
Yes, the mucopolysaccharidoses, or MPS, are a profoundly impactful group of hereditary diseases. They affect roughly 1 in 25,000 live births, so they're considered rare, but devastating for those affected.
Right.
They're directly caused by a genetic deficiency in any one of the specific lysosomal hydrolases normally involved in meticulously breaking down heparan sulfate and/or dermatan sulfate.
Okay, specific GAGs.
Specific GAGs, yes. These are progressive disorders, meaning the symptoms sadly worsen over time as the undegraded material keeps accumulating. They're characterized by that pathological buildup of partially degraded GAGs inside the lysosomes of various tissues throughout the body.
And that causes problems everywhere.
Pretty much. This accumulation leads to a wide spectrum of symptoms, noticeable skeletal abnormalities, deformities of the extracellular matrix, and in many cases, severe intellectual disability.
How are they inherited?
Most MPS disorders are autosomal recessive, meaning you need to inherit 2 copies of the defective gene, one from each parent, to get the condition.
Okay.
The significant exception is Hunter syndrome, which is X-linked. The defective gene is on the X chromosome, so it impacts males more frequently and severely.
Right.
A striking and particularly tragic aspect is that children born homozygous with 2 copies of the affected gene usually appear normal at birth.
Really? Normal at first?
Yes, but then they gradually deteriorate as the undegraded GAGs progressively accumulate in their tissues, impacting organ function. In severe cases, these deficiencies can unfortunately lead to death in childhood. It's heartbreaking. Research is constantly ongoing for better treatments.
How are they diagnosed then, if kids seem normal initially?
Diagnosis often starts with finding oligosaccharides, those partially degraded GAG fragments in the urine. Identifying the specific structure on the non-reducing end of these fragments can be a powerful clue.
Because that's the bit the missing enzyme should have removed.
Exactly. It points to the specific enzyme deficiency. The diagnosis is then definitively confirmed by measuring the patient's cellular levels of the suspected lysosomal hydrolases using specialized assays.
Are there treatments?
Well, there's no definitive cure currently, but some treatments have shown limited success. Bone marrow and cord blood transplants have been tried. The idea is transplanted healthy macrophages can produce the missing enzymes.
Does that work well?
It has variable success. Enzyme replacement therapy, EART, is also available for some types, like Hurler and Hunter syndromes. But, and this is crucial for mastery, while ET can help with some systemic symptoms like joint mobility or organ size, it typically does not prevent the neurological damage. The large enzyme molecules often can't effectively cross the blood-brain barrier to get into the central nervous system. That remains a major challenge.
I see. Can you give some key examples? Hurler, Hunter?
Sure. These are important to know. Hurler syndrome, MPSI, caused by a deficiency in iodide, degrades dermatan and heparin sulfate, often characterized by corneal clouding, skeletal deformities, intellectual disability.
No.
Hunter syndrome, MPS II. X-linked deficiency in aduronate sulfatase, also degrades heparin and dermatan sulfate. Shares features with Hurler, but usually less severe, no corneal clouding.
Got it.
Morquio syndrome,
Right.
MPS-IV, types A and B. Deficiencies in enzymes degrading keratan sulfate, galactosamine 6-sulfatase or VOG-galactosidase. Leads to severe skeletal issues, but often spared intellectual function.
Right.
Merito-Lamy syndrome, MPS 6. Deficiency in aryl sulfatase B degrades dermatan sulfate. Skeletal deformities, short stature, coarse facial features, but intellect is generally normal.
Okay, so different enzyme, different GAGs affected different symptoms.
Precisely. And then there's a particularly rare and severe one called multiple sulfatase deficiency, or Austin disease.
Multiple sulfatases, all of them.
Yes, in this devastating condition, all sulfatases in the body are non-functional. It's due to a defect in forming formylglycine, an amino acid derivative needed for these enzymes to work at all.
Wow. One small defect affects a whole class of enzymes.
Exactly. It highlights how a single fundamental defect in a common modification pathway can cripple an entire class of enzymes, leading to profound, far-reaching consequences across multiple GAG degradation pathways.
That's a powerful illustration of how critical breakdown is and how one missing enzyme can have such widespread devastating effects. Okay, let's pivot now to glycoproteins. You call them the sophisticated cousins of proteoglycans.
Yes, that's one way to think of them.
They're also carbohydrate-protein combinations, but you said they're more about specific, intricate signals, right? Less about bulk, more about cell communication and identity.
Precisely. Glycoproteins represent another vast and incredibly important class of biomolecules. They are defined simply as proteins that have oligosaccharides, often called glycans, covalently attached to them. And glycosylation, the process of adding these sugar chains, is actually the most common post-translational modification of proteins in our bodies.
Most common. Wow, so it happens a lot.
It happens everywhere, underscoring its widespread importance across virtually all cellular processes. Now, they differ from proteoglycans in several key ways, which
Cool.
are important to grasp for a nuanced understanding for that mastery level.
Okay, key differences. What are they?
First, carbohydrate content. While proteoglycans can be up to 95% carbohydrate, glycoproteins usually have much less. Highly variable, but typically less. For instance, IgG, an antibody,
The right immune system.
is less than 4% carbohydrate by mass. Compare that to aggrecan, the proteoglycan, at over 80%. Big difference.
Huge difference. Okay, what else?
Second, the glycerine structure itself is distinct. In glycoproteins, it's relatively short, usually 2 to 10 sugar residues, maybe longer sometimes. Crucially, they do not have those repeating disaccharide units like GAGs.
No repeating units. Different structure.
Very different. Glycoprotein glycans are structurally much more diverse. They're often highly branched, like little trees, instead of being linear, like GAGs. And they may or may not carry a negative charge, depending on whether they have acidic sugars, like nana.
So shorter branched, more diverse sugar chains.
Exactly. Despite their generally smaller carbohydrate part, glycoproteins participate in an incredibly broad range of cellular phenomena. They effectively act as the cell's identification tags and communication antenna.
ID tags and antenna, I like that.
They're fundamental to cell surface recognition, allowing cells to identify and interact with other cells, with hormones carrying messages, even with viruses trying to invade. They contribute significantly to cell surface antigenicity. A classic, highly relevant example is the ABO blood group antigens on red blood cells. Those are distinct glycoprotein determinants.
So glycoproteins determine blood type.
The carbohydrate parts of specific glycoproteins, yes. They're also important components of the ECM, helping integrate cells into their surroundings, and they form key components of mucins.
Mucins, like mucus.
Yes, those slippery, gel-forming proteins in the GI tract, the urogenital tracts. They act as protective biological lubricants, trapping pathogens, protecting delicate tissues.
Got it.
And notably, almost all the globular proteins in human plasma, with the major exception of albumin, are actually glycoproteins.
Really? Most plasma proteins.
Most of them, yeah. Highlighting their ubiquitous presence and diverse roles everywhere, from transport to immunity.
So their oligosaccharide components might be smaller than GAGs, but they're incredibly diverse and crucial for cell identity and signaling. That blood type example really hammers home the specificity. Tell us more about the structure of these oligosaccharides and how they actually link to the protein. Are there different ways the sugar attaches?
Absolutely. The structural diversity of these oligosaccharides is precisely what gives them their incredible signaling power. The oligosaccharide parts of glycoproteins are generally branched heteropolymers.
Branched again? Okay.
Made of different kinds of sugars, arranged in complex tree-like structures. This branching, combined with the wide variety of sugars they can contain, allows for immense structural diversity. It creates a vast molecular language.
A language of sugars.
Pretty much. They're primarily composed of dehexases, 6-carbon sugars like glucose or galactose. Sometimes you'll find neuraminic acid, that 9-carbon nose, and L-fucose, a 6-deoxyhexose, adding further complexity and specificity.
Okay. How do they attach to the protein?
The attachment is also very specific. It happens through one of two main types of stable glycosidic links. First, there's the N-glycosidic link. Here, the sugar chain attaches to the amide group of an asparagine side chain within the protein. The N stands for the nitrogen in that amide group. Very common, very important linkage.
Okay, N-link to asparagine. What's the other?
Second, you have the O-glycosidic link.
Like.
In this case, the sugar chain attaches to the hydroxyl group of either a serine or a threonine side chain in the protein. The O stands for the oxygen in that hydroxyl group.
O-link to serine or threonine. Got it.
And just to note, there's a special O linkage specifically in collagen between galactose or glucose and the hydroxyl group of hydroxylysine, a unique modification for collagen's structure.
Okay, so N-linked or O-linked.
Yeah.
Can a protein have both?
Yes. What's really clever is that a single glycoprotein molecule can contain only N-links, only O-links, or it can have both types within the same molecule.
Wow, okay, that adds even more complexity.
It does. This dual attachment capability means the cell has an even wider palette to create incredibly diverse and specific molecular signals. Like having two different alphabets to write complex molecular messages, enhancing their functional versatility immensely.
Let's break down the N and O-link types a bit more then. What are the differences in their sugar chains?
Good idea. O-linked oligosaccharides can be quite varied structurally. One or more sugars arranged in linear or branched patterns. Many are found in extracellular glycoproteins, or as components of membrane glycoproteins, interacting with the outside world.
Like the blood type ones?
Exactly. The ABO blood group determinants on red blood cells are a classic example of O-linked glycans. If the terminal sugar is N-acetylgalactosamine, GalNAc, it's blood group A.
Right.
If the terminal sugar is galactose, it's blood group B. And if neither GalNAc nor galactose is the terminal sugar, it's blood group O.
Such a small difference, such huge implications.
Absolutely. Shows how molecular specificity dictates biological function. Now, N-linked oligosaccharides, they fall into two broad classes: complex oligosaccharides and high-mannose oligosaccharides.
Complex and high-mannose.
Okay. Despite their differences in final form, both classes share the exact same pentasaccharide core structure.
A common core.
Yes, a 5-sugar core. It provides a foundational scaffold, a common starting point. Where they diverge is in the additional sugars attached to this core. Complex oligosaccharides have a diverse group of other sugars added: GLCNAc, GalNAc, L-fucose, NANA, creating highly branched, varied structures.
Okay, and high-mannose.
High-mannose oligosaccharides, as the name suggests, primarily contain more mannose residues beyond that core. This structural diversity, starting from a common core but with different branches, allows for a vast array of recognition signals on the cell surface, a sophisticated molecular language between cells and their environment.
It's astounding how specific these sugar additions are, dictating something as fundamental as blood type. Really underscores the precision of the cellular machinery. Yeah. Now, let's talk about that cellular assembly line. How does the cell actually craft these sophisticated glycoproteins? It sounds like an even more highly precise, orchestrated process than GAG synthesis, given their signaling roles.
It absolutely is. The synthesis of glycoproteins is another marvel of cellular engineering. Incredible precision, sophisticated division of labor within the cell. Now, proteins destined just for the cytoplasm are typically made on free ribosomes out in the cytosol.
Right.
But the vast majority of glycoproteins, those heading for membranes, lysosomes, or export out of the cell, are synthesized on ribosomes specifically attached to the rough endoplasmic reticulum, the RER.
Attached to the RER again.
Yes. These proteins contain specific signal sequences like molecular addresses or zip codes, ensuring they go to the right place. An N-terminal hydrophobic sequence guides them to the RER, and the growing polypeptide chain gets extruded right into the RER lumen, the interior space.
Okay, then what?
From the RER, they're transported via specialized secretory vesicles to the Golgi apparatus.
The sorting center.
The sorting center, exactly. In the Golgi, the glycoproteins undergo further modification, and then they're meticulously sorted based on their final destination. Those meant to be secreted or targeted to lysosomes get packaged into specific vesicles. These vesicles then fuse either with the cell membrane to release contents outside or with the lysosomal membrane to deliver enzymes inside.
What about the ones for the cell membrane?
Right. For glycoproteins meant to be permanent components of the cell membrane itself, they get integrated directly into the Golgi membrane. This modified Golgi membrane then buds off, forming vesicles that ultimately fuse with the cell membrane, adding their glycoproteins to the cell surface.
Okay.
And a key detail here for functional understanding. These membrane glycoproteins are oriented with their carbohydrate portion facing the outside of the cell.
Facing outwards. Why?
Makes them readily available for crucial cell surface recognition and interaction with other cells or molecules in the extracellular environment. That's where the signaling happens.
Ah, makes sense. Okay, what about the sugar building blocks for these glycoproteins? Same ones as for GAGs?
Mostly the same activated nucleotide sugars, yes. UDP glucose, UDP galactose, UDP GL-CNAC, UDP GalNAc.
Okay.
Plus other important donors like GDP mannose, GDP L-fucose, which is made from GDP mannose, and that unique one, CMP nana.
Right, the monophosphate one.
Exactly. And remember, when acidic nana is present, it gives the oligosaccharide a negative charge at physiological pH, which can be important for interactions. These oligosaccharides get covalently attached to specific amino acid side chains. The crucial factor determining where glycosylation occurs is the protein's intricate 3D structure.
The protein shape dictates where the sugars go.
Precisely. The shape determines if a particular amino acid residue is accessible and recognized by the glycosylating enzymes.
Okay. So how does the synthesis differ between O-linked and N-linked? Let's start with O-linked.
Right. O-linked glycoprotein synthesis. It shares significant similarities with GAG synthesis, but obviously uses different sugar chains. First, the core protein is made on the RER, extruded into the lumen.
Okay.
Glycosylation then begins with the transfer of N-acetylgalactosamine, GalNAc, from UDP GalNAc, directly to the hydroxyl group of specific serine or threonine residues on the protein.
Directly onto the protein.
Directly onto the protein, yes. The enzymes responsible, glycosyltransferases, are typically membrane-bound in the Golgi. They carry out the stepwise synthesis, adding individual sugars one by one. Stepwise. And what's truly remarkable, as we hinted at, is that these enzymes act in a very specific order without needing a template, unlike DNA, RNA, or protein synthesis.
Naito template. How do they know what to add next?
They recognize the actual 3D structure of the growing oligosaccharide chain itself as the appropriate substrate. So the protein's conformation and the growing sugar chain act as the template for the next addition. It ensures the correct sequence and linkage.
That's a profound difference. No template, just recognizing the growing chain. That implies an extraordinary level of molecular recognition. So how does N-linked glycoprotein synthesis differ then? I know dolichol is a key player there, which suggests a whole different strategy.
Indeed. N-linked glycoprotein synthesis has its own entirely unique and equally fascinating mechanism. It primarily occurs in the lumen of the RER, and it definitely involves that crucial lipid dolichol.
Dolichol, right.
Dolichol is a phosphorylated form of a long-chain lipid, specifically dolichol pyrophosphate, and it's embedded right there in the RER membrane. The initial glycoprotein product then undergoes further processing, trimming in both the RER and Golgi before reaching its final form.
Okay, so dolichol is involved from the start in the RER. How?
Unlike O-linked, where individual sugars are added directly to the protein, N-linked glycoproteins don't get glycosylated with individual sugars directly onto the protein itself. Instead, a large preformed lipid-linked oligosaccharide is first constructed.
Deformed on what?
On dolicle. This complex oligosaccharide is built right on the dolicle carrier in the RER membrane, attached via a high-energy pyrophosphate linkage.
Okay, a sugar chain built on a lipid first. What sugars are in it?
It's a very specific structure. Typically contains 2 N-acetylglucosamine, GLC-N-saccharide residues, 9 mannose residues, and 3 glucose residues. A 14-sugar unit in total.
14 sugars.
Yeah.
Built on dolicle.
Exactly. These sugars are sequentially added to the dolicle by various membrane-bound glycosyl transferases, starting with GLC-NEC, then mannose and glucose. Once this entire 14-sugar oligosaccharide is complete on the dolicle carrier, it is then transferred in one single enzymatic step from dolicle to the ematted nitrogen of a specific asparagine residue within the protein that's being glycosylated.
Transferred all at once.
All at once. Catalyzed by a protein oligosaccharide transferase enzyme, also in the RER. As a side note, an antibiotic called tunicamycin specifically inhibits this N-linked pathway by stopping that initial transfer of GLC-NAC to dolicle.
Useful tool for studying it, I guess.
Exactly. And clinically, this pathway is critical. Congenital disorders of glycosylation, CDGs, are severe syndromes caused primarily by genetic defects in N-linked glycosylation, either in assembling the oligosaccharide on dolichol, type I CDGs, or in processing it later, type 2 CDGs.
Okay, so the 14 sugar block gets transferred. Is that it then?
Not quite. After this initial transfer, the N-linked oligosaccharide isn't finished. It undergoes crucial processing like quality control and refinement. As the glycoprotein moves through the RER, specific mannose and glucosyl residues are strategically trimmed off by enzymes.
Trimmed? Why remove sugars you just added?
It's part of the maturation and quality control process, ensuring proper folding and signaling. Then, as the glycoprotein journeys from the RER to the Golgi, the oligosaccharide chains are completed by adding a wide variety of other sugars, GLC-NAC, galanac, more mannoses, then fucose or nana, often as terminal groups.
So trimming and then adding different things back.
Right. This comprehensive set of additions leads to the formation of what we call complex glycoproteins with those diverse branched sugar chains.
Okay. What was the other type? High mannose.
Alternatively, some N-linked oligosaccharides might not undergo this extensive further processing. They just keep their initial structure after trimming, remaining as branched mannose-containing chains. Those result in high mannose glycoproteins.
Got it. Complex or high mannose?
The ultimate fate of these N-linked glycoproteins is similar to O-linked ones, secreted, cell membrane, or targeted to lysosomes. And it's vital, again, to distinguish this precise enzymatic glycosylation from non-enzymatic glycosylation called glycation.
Glycation. That's the random sugar sticking.
Yes, the random, uncontrolled attachment of sugars often linked to chronic conditions like diabetes. Very different from the controlled enzymatic process we've been discussing. Essential distinction for mastery.
That's an incredibly detailed assembly line. From the RER, using lipid carriers, trimming, adding back.
Yeah.
Building these molecules is clearly a highly controlled dance. But just like GAGs, glycoproteins also need to be efficiently broken down right when they're old or damaged.
Absolutely. Turnover is constant.
And sometimes the problem isn't the breakdown itself, but getting the tools for break down the enzymes to the right place. This leads us to a really impactful and, well, perplexing disease, doesn't it?
You're absolutely right. The degradation of glycoproteins is critical for cellular health, and it's quite similar in principle to lysosomal GAG degradation.
Back to the lysosomes again.
Back to the lysosomes, it employs that same arsenal of lysosomal acid hydrolases. Each enzyme is generally specific for removing one particular sugar or component of the glycoprotein.
Specific tools for specific jobs.
Exactly. They are primarily exoenzymes, removing groups one by one from the non-reducing end, typically in reverse order of incorporation. That last on, first off rule applies here too.
Last on, first off again.
Okay. This precise sequential removal is crucial because if any single degradative enzyme is missing or non-functional due to a genetic defect,
the whole process stops.
The entire degradation pathway by the other exoenzymes comes to a screeching halt. This leads to a buildup of partially degraded glycoprotein structures within the lysosomes.
Causing problems, presumably.
Causing significant cellular and tissue damage. This leads to a group of very rare autosomal recessive diseases called glycoprotein storage diseases, or oligosaccharidoses.
Glycosaccaridosis, okay.
They're caused by a deficiency in one of these specific degradative enzymes, resulting in harmful accumulation of partially degraded glycoprotein structures in lysosomes. A classic example is amanosidosis type 3.
Mannosidosis, missing a mannose-removing enzyme.
Exactly. Deficiency of amanosidase. It's severe, progressive, often fatal. Its presentation can look similar to Hurler syndrome with skeletal and developmental issues, but it uniquely includes immune deficiency. Diagnosis often involves finding mannose-rich oligosaccharide fragments in the urine, confirmed by an enzyme assay.
Okay. Now, you mentioned enzyme targeting. Getting the tools to the right place, that's where eye cell disease comes in.
Precisely. This is where the story gets even more complex and critical, and it leads us straight to the mystery of eye cell disease, a fascinating and vital aspect of N-linked glycoproteins, particularly those destined for the lysosome itself, is how they are specifically marked for delivery. While being processed in the Golgi, these N-linked glycoproteins can be phosphorylated.
Phosphorylated, a phosphate group added.
Yes, a phosphate group is added, specifically on carbon-6 of one or more of their mannose residues. This phosphorylation acts as a crucial molecular tag, like a specific postal code.
A postal code for the lysosome.
Exactly. The phosphate itself is provided by UDP-GLCNEC in a reaction catalyzed by a very specific phosphotransferase enzyme. Once tagged with this mannose 6-phosphate, M6T, specific receptors located in the Golgi membrane recognize and bind with high affinity to these M6P residues on the proteins. This binding acts as a molecular sorting signal. These tagged proteins are then packaged into specialized vesicles that bud off from the Golgi and are precisely sent to the lysosomes. It ensures the lysosomes receive their necessary supply of degradative enzymes. It's an incredibly efficient cellular postal service.
Wow. Okay, so what goes wrong in eye cell disease?
This brings us directly to eye cell disease, also known as mucolipidosis type 2. It's a rare but devastating lysosomal storage disease. The fundamental defect in eye cell disease is a genetic deficiency of that crucial phosphotransferase enzyme.
The enzyme that adds the M6P tag.
The very one. Because this enzyme is deficient, the acid hydrolases the essential enzymes needed for lysosomal degradation of GAGs, glycoproteins, everything else.
Yeah.
They cannot be properly phosphorylated with the mannose 6-phosphate tag. Consequently, these essential enzymes are not correctly recognized by the Golgi receptors, and instead of being targeted to the lysosomes,
She goes somewhere else.
they get mistakenly secreted out of the cell into the bloodstream.
Secreted, but they're needed inside the lysosome.
Exactly. The result is profound and tragic. The acid hydrolases that should be abundant and active inside the lysosomes are conspicuously absent or severely deficient there. This leads to a massive accumulation of various undegraded substrates, GAGs, glycoproteins, lipids, causing the lysosomes to swell up and form large, distinctive inclusion bodies.
The inclusion cell name, eye cell.
Precisely why it's called eye cell disease. Because of these characteristic inclusions seen in patient cells, clinically, patients present with severe symptoms. Significant skeletal abnormalities, severely restricted joint movement, coarse or dysmorphic facial features, profound psychomotor impairment.
And how is it diagnosed differently from, say, MPS?
A key diagnostic indicator that distinguishes eye cell disease is finding high amounts of lysosomal enzymes in the patient's plasma and urine.
High levels outside the cell.
Yes. This indicates the problem isn't a lack of synthesis of these enzymes, but a profound defect in their targeting mechanism to the lysosomes. They're being made, just sent to the wrong address.
I see. That's a critical difference.
It is. Eye cell disease shares features with both mucopolysaccharidoses and sphingolipidosis, so it's classified as a mucolipidosis type 2. Unfortunately, there's currently no cure, and affected children typically succumb to cardiopulmonary complications early in childhood. The key distinction to really nail down for mastery is this. While MPS involves defects in the JAG degradation enzymes themselves, eye cell disease involves a primary defect in the targeting mechanism that delivers those essential lysosomal enzymes in the first place. It's a failure of the cellular postal service, leading to much broader dysfunction.
So from the incredibly slippery consistency of our mucus to the intricate dance of cell recognition, and even the devastating consequences when cellular postal codes go awry, these unsung heroes, GAGs, Proteoglycans, and glycoproteins are constantly at work, often completely unnoticed.
Absolutely, underlying so much.
We've gone from their fundamental negative charges and water-binding properties to the precise molecular assembly lines that build them, and then to the specific molecular defects that can lead to these profound and heartbreaking diseases. I think you listening now have a truly solid, comprehensive understanding of a field that is absolutely critical to our basic biology and, well, to our very existence.
And if we connect this to the bigger picture, understanding these molecular building blocks isn't just, an academic exercise. It's the very foundation for developing new diagnostic tools, revolutionary therapies for inherited disorders like MPS and eye cell disease.
Right, practical applications.
Exactly. It's also absolutely crucial for cutting-edge fields like regenerative medicine and tissue engineering, where scientists are trying to grow and repair complex tissues. These molecules are vital for creating the right environment, the precise scaffolding for cells to thrive and form functional tissues. So, this deep dive into their complexity, therefore, raises a really important question for the future, doesn't it? How might our growing mastery of these incredibly intricate biomolecules allow us to design even more sophisticated interventions for health and healing? Could we potentially correct these defects with unprecedented precision, or even build new, perfectly functional tissues to replace damaged ones down the line?
That's a powerful thought to leave with. The idea that understanding these microscopic worlds so deeply can unlock macroscopic solutions for human health. Something for all of you to mull over until our next deep dive. Stay curious.